Hi, everybody. All right, so we're in the middle talking about different types of genetic technologies. That is, things that scientists have done to alter the natural way that genes are used in organisms. And the first thing that we talked about was genetic engineering, which takes a gene from one species and puts it in another. That creates new individuals with new traits that have never been seen before. In one very real sense, that increases variation. Today, we're going to talk about a technology, a genetic technology that's essentially the opposite. This takes an individual and makes an exact clone, an identical copy. Now, we've talked about something in nature that makes exact copies. Type of cell division that makes exact copies is mitosis. Mitosis copies all the DNA in the cell, in the nucleus, and then divvies up very carefully to make sure each end of the cell gets an exact copy, and then that cell divides and you have two identical cells. And if this is an organism growing, then it'll copy the DNA and divide it and you get 4 identical cells and so on. Well, scientists have figured out how to use that process for cloning. Now, many organisms on the planet clone naturally. Plants. If you make a cutting of a plant, right? We talked about this asexual reproduction. You can take the cutting and put it in water and it will start to divide by mitosis and make roots and then grow into an identical, genetically identical organism. Cloning. Well, their plants will clone, but animals, well, at least mammals, vertebrates do not clone naturally. But scientists have figured out how to do it. Well, how do you do it? Well, imagine, let's say for the moment that we wanted to clone me. Don't want to do that, but let's just pretend for the moment. Where might you find the information for how to make me? A nucleus of any cell in my body that has a nucleus, and that's the vast majority of them. So we could take a cell, right, and take the nucleus, and that has the full instructions how to make me. All that DNA can be transcribed into mRNA and into proteins that are Ms. Cohen-Norris, right? So what if we could take that information and put it in a cell that's able to grow into a whole new baby? What kind of cell grows by mitosis into a whole new baby? A zygote. So if we could take my DNA, which is copies of what I got from half from my dad and half from my mom, and we could put that into a zygote and make that zygote grow into a baby by mitosis, you're going to get a baby that is genetically identical to me. You'll get a little baby Ms. Cohen-Norris. Now, hopefully, nobody ever clones a human being, but we have cloned mammals. We've cloned a sheep by doing exactly that. The DNA from the adult is put into a zygote. That zygote divides by mitosis. And when that individual grows, it will be identical to the original, the adult donor, where the nucleus came from. So this is how it works. If you take a look here, you have a donor cell comes from this sheep, and we get a zygote from this sheep, and we combine them. Okay, we can take that egg and put in the nucleus from this donor sheep, give it all its DNA for how to make it, put it in that zygote, and let it divide by mitosis and grow and grow and grow and grow. Put it in the uterus of another sheep, and let that sheep carry it for as long as sheep are pregnant and have a baby right there. So if this baby is compared, we can compare this baby to the sheep where the DNA came from, or the sheep that gave the egg zygote, or the sheep that was pregnant. Which sheep is it going to be identical to? The one that gave the DNA. So we have the nucleus from the donor sheep, and that's got all the DNA that ultimately is going to be here that's going to grow into the cloned baby. Get the egg cell right here. We're going to combine them, and it's going to grow from the zygote into an embryo, and then ultimately into a cloned baby that's identical to the original donor.